Welcome to another episode of Progress, Potential, and Possibilities, discussions with fascinating people designing a better tomorrow for all of us. I'm your host, Ira Pastor.
Welcome, everybody, again to another episode of our show. Today, bringing you another really fascinating guest involved in creating a better tomorrow on many unique fronts. Today, we have the honor of being joined by Dr. Seemay Chou, who is the co-founder, Chief Executive Officer, and board member of Arcadia Science, which is a really interesting research and development company that's focused on under-researched areas in biology with a very specific focus on novel model organisms that haven't been traditionally studied in a laboratory, all ultimately with the goals of unlocking some of this real interesting knowledge and ingenuity contained within a wide range of these diverse species, uncovering how evolution has solved a lot of problems in the past that we deal with as humans today, and ultimately revealing this untapped biologic innovation, helping to generate new technologies, products, and so forth. Dr. Chou is also the co-founder of a really interesting company called Trove Biolabs, a startup that's focused on harnessing novel bioactive moieties found in tick saliva for skin therapies. Dr. Chou joined and set up Arcadia. Prior to that, she was at UCSF as an assistant professor in biochemistry and biophysics. She received her Ph.D. in molecular cell biology from UC Berkeley. She did her bachelor's in cellular molecular biology, University of Texas at Austin. A lot of really interesting themes to get into today with her. Honored to have her. Dr. Seemay Chou, welcome to our show. Thanks for joining us.
Awesome. Thanks so much. Excited to chat with you.
Yeah, it's a really exciting set of topics. I would love to start off just by handing you the floor for a little bit. If you could take us sort of a little bit into the early days and then sort of your interest in molecular and cellular biology, but then sort of this general theme that we'll be coming back to a lot in the show in terms of learning from nature. You've published on, a variety of what I'll call critters, everything from looking at antiviral compounds and sea anemones, the morphodynamics of neural tube patterning and sea squirts. We'll get into ticks in a little bit. What got you interested in sort of this general theme of sort of looking at all the things that nature has already solved and sort of applying them for human health?
Yeah, I think basically in my career, I really jumped around between a lot of different topics because at the end of the day, I love to tinker. And I think my really the feeling that drives me is just it feels like such a privilege to basically tinker with nature and learn how these things have already been solved. I mean, it's just incredible, it's really like humbling because you sit there and you try and tinker and figure out an intelligent way of solving different things. And then when you get some insight into how it's already been done, you just feel so small, And I think it's a wonderful feeling actually, because it's just like incredible. So I think I'm, maybe in simple terms, just like a big fan of biology. And I think that I just want more of that. And we're trying to figure out how to do this in a slightly more sort of engineering way. How can we actually engineer more of these moments and capitalize on them to solve actual problems in the world?
Excellent. And it's very interesting thinking about that theme of engineering. I come out of the pharmaceutical industry, and that industry has an amazing sort of 100-year plus history of going to nature, primarily looking at, small molecules from bacteria and fungi and plants and so forth, which has yielded the industry that we have today. But you're taking sort of a much more comprehensive look at things, in the sense that you're not just looking at sort of, hey, what secondary metabolites do we have sitting around here? You look at the host microbe interactions. You look at symbiosis. A lot of these themes that have not really been touched on, I sort of put them in this category of interactome, which is sort of one of the themes that's on your UCSF site. Talk a little bit about this, because I think this is important in terms of, look, and we'll get into Arcadia in a little bit, but really looking at different tools that we have available in now, as we get close to 2023, and going behind sort of like, the old grinding and finding approaches of yesterday towards these more sort of integrated understandings of what these moieties are all about, how they work, and why they're there in the first place.
Yeah, I mean, I think so. So basically, like over my many years of being a scientist, like always the most fascinating things that I've learned have been things that I haven't been looking for. They've just been kind of like surprises. And I'll give you like one example is, I was studying these toxins that get exchanged between bacteria when they actually fight each other. So interbacterial warfare. And this happens a lot. They sort of like sharpen their tools to defend against other microbes around them. And it turned out, as we started poking around in them, I was originally trying to understand the mechanism of those, but we found that the genes that encoded those had been horizontally stolen by a number of eukaryotes to basically repurpose them for a whole nother function in the biology of a different set of animals. And I just like, this was so cool. And it like was never something that I could have known to look for, at least in the perspective I was coming in, but actually gave like huge insights into just the importance of this toolkit and all the different ways that it could be used. And like the fact that it was so important that it could be stolen in this way horizontally. And so I think like that's just one example of so many where I'm looking in one direction and like something way cooler smacks me. And I think what I've been trying to figure out for a while now is like how we engineer these moments of serendipity rather than just letting them happen to us so passively. Because I mean, the truth is like biology has figured out a lot of these things. And like, I just don't know how we like learn about it with a little more intention. And a lot, like let's look at antibiotics. That whole field opened up because of something that happened where accidentally we found these things from fungus that kill bacteria. So I think like at a more conceptual level, this is like one of the big problems I'm trying to solve through Arcadia is like, how do we even learn how to learn? Like if you accept the originating hypothesis that there's a ton of innovation across the tree of life, like how do we navigate it? How do we navigate it in a way that it's not just surprises, And how do we navigate it from first principles of like, here is X problem I'm trying to solve? Like, how do I explore the tree to figure out the best places to look rather than saying like, okay, I'm starting with this system and I'm going to milk everything I can out of it, which is a perfectly valid approach, by the way. I'm not at all dissing that. I think we need that. But then we don't have the other approach quite figured out yet, sort of systematically exploring innovation across the tree. So that's kind of the basic thing that we're trying to figure out for now. We haven't, like decided on which organisms exactly we're going to double down on yet, because I think we've run into this paralysis of like, how do you choose? Right. And so we're still first trying to just like hone our skills in thinking about that question. And then, I think like, the big shortcut for a lot of the like commercial sort of goals that are out there is like to see where it's already been solved. It's been battle tested in evolution. And so like if you were, let's take, for example, like, a lot of drugs are about cellular delivery. And so like you actually have an enormous amount of diversity across the tree of life of like how things get in and out of cells, the capacity for that being regulated. So if that were your goal from an evolutionary biologist perspective, like how might you sort of systematically try and understand that? So I don't know does that answer your question? Absolutely. I'm happy to like comment with a little more concreteness too.
No.
Yeah.
It was spot on, and I'm glad you brought up the sort of the tree of life there, because we'll go to different parts of that tree, throughout our discussion. And I think, obviously a great place to begin would be with the ticks, and I'm not sure which part of the tree of life they ended up in. But if we look at this theme, and we actually touched on it a little bit in the past, this sort of hematophagous organisms in the case of ticks, and we've, on past shows we talked about mosquitoes and leeches and things like that. And obviously organisms that love to suck on blood, whether it's human blood or the blood of other organisms, don't like other stuff happening. They don't like coagulation. They don't like an immune response. They don't like inflammation. They don't like a bunch of stuff that happens when they do that. And evolution, has given this amazing toolkit to produce anticoagulants and anti-inflammatories and all sorts of cool things like that. Take us a little bit into that world, because before Arcadia, sort of the beginning of all this is Trove Biolabs and sort of this first stop in the tree. But at the same time, if you could talk a little bit about some of the work you were doing, because you published a lot on, again, not just the ticks' biochemical warfare capabilities, but also the synergy with some of the microbes that sort of got them, over time, allowed them to evolve these capabilities, especially when it comes to the antimicrobials and stuff like that. Take us down both those paths, if you would.
So I basically landed in ticks on complete accident. I never, ever intended to study ticks for more than maybe 6 months. It was because I was actually studying this bacterial warfare thing, and we found this horizontal gene transfer. So, okay, all of a sudden now we have these toxins encoded in tick genomes. Yep. And so I thought, oh, this will be an easy side project. I'll just stop off for a second, knock these down in ticks, show that there's an effect, move on. But I think what I started becoming obsessed with after that is it's once you start pulling this thread, you realize there's a giant ball of yarn of connections, and it's not so simple as figure 4 of a paper, that you really have to think about this well. You have to really understand the biology of the organism, and not just through your sort of anthropomorphic viewpoint as a human.
Yeah.
So maybe the specifics might matter a little bit here. So I'll just kind of talk through it a tad.
Take your time.
So originally the hypothesis was, okay, ticks have this gene, it's antibacterial. The easy place to start is let's just knock it out and show that it has some effect on a well-known microbe that ticks carry, which is the Lyme pathogen, this bacterium, Borrelia burgdorferi. Okay, so we'll knock it down, we'll see some effect, go home. But I think what that experience got me to understand, of starting to really delve into this, is that is such a human perspective, to think that the pathogen that ticks carry that infect us would be the target of a tick's immune system. In fact, that's the complete opposite of what's going on. The fact that they can carry this microbe stably and infect us is actually evidence that they have a stable interaction with this microbe. If Borrelia were deadly upon contact with ticks, and they had to defend fiercely against this, it would not continue to be a stable interaction, right? We are the problem. We are the problem because we are not the primary preferred host of ticks, and so we run into this microbe by accident. But if you exclude us from that circle of life, it has its own preferred hosts, and it's actually all these organisms living in harmony with each other. And so that was really cool. I mean, it seems so obvious now, but back, it took me a while to get there because of the perspective I was coming into this. So that is a moment where you start thinking let me stop trying to overlay my problems and my perspective onto the system, and just try and understand how sort of awesomely beautiful it is that the system exists without me. And why do these microbes want to live in the tick? Why do the ticks want to partner with the microbes? There's something harmonious going on here. It's a partnership. And so originally I was very focused on how do we sort of disrupt, just very narrowly focus on disrupting Lyme, which I'm not saying is a bad goal, but it's a very specific goal that sometimes you kind of miss the broader biological context here. What you're trying to disrupt is a symbiotic partnership. And so trying to understand why it's dependent on the tick and why this might exist was helpful. And then that's what led me down the path of saliva. because there's these studies that have been done like decades ago showing that, the tick saliva itself is really important for the Lyme pathogen to survive and spread when it gets into us, and that there's like a literal symbiosis there with the molecules in the tick saliva. So if you just take Borrelia and you inject it into your skin, its survival rate is quite low. But if you co-inject it with tick saliva, survival rate goes up and it spreads better in your body. So then then you keep pulling that thread, like, okay, like, so why did it choose the tick? And of course I'm anthropomorphizing again, but like, there's something really special about ticks here, like the fact that they feed for so long means that they have a completely different set of molecules that they're deploying. They have to deal with the temporal issue of sort of like short-term versus medium and long-term events that happen in the skin. And actually it makes a ton of sense, right? Like it's consistent with the idea of like, for some pathogens, those activities are so helpful for them to complete their infectious cycle. So I think like I spent a lot of time thinking about like, how could I have like basically landed on this trove idea sooner? Like if I hadn't been spending all this time, like limiting my understanding to this, like one specific sort of very human-centric view, like maybe I actually would have gotten there sooner. So with Trove, what we're doing is like taking a huge step back and just being like, okay, microbes figured this out before us. Ticks are like awesome dermatologists and they've evolved all this stuff in their saliva. Like what else is there that we haven't discovered? And I think, like, it sounds like you've talked about this before on your show, but like, yeah, mosquitoes and leeches and all these other blood sucking organisms, like they have different tactics. And that is like so cool in itself. And blood feeding evolved many independent times over evolution.
Yep.
And so that's kind of the history of just like how we got from like thinking about intermicrobial warfare to this, but how do we speed that up? Like, and some of it is just like my frustration with myself of like, how did I not see this sooner, but there are also like real blocks, I think, like in terms of like incentives and tools and stuff that also slow this down a bit.
Sure.
Yeah.
As we think just if we stay on the tick for a moment, we think about sort of the pathogen interactions and so forth. One of the interesting themes there that pops up, actually it pops up in a part of Arcadia's work as well, but is this topic of quorum sensing and anti-virulence factors and so forth. And I bring that up because thinking back to sort of some of my old days in this, nature doesn't make, you're just pointing out with penicillin, so they don't just make one thing, right? Typically
right.
These organisms make these amazing cocktails of stuff, and it's not always like they don't always make a great antibiotic. In the case of quorum sensing compounds, they make the compounds so the microbes don't stick together, they don't kill them, but it makes life uncomfortable for them. Are you running into any, and if this is confidential, obviously don't go into it, any interesting mechanisms as you're starting to study some of these bioactive antimicrobials that like, wow, okay, this might not be, great at killing XYZ, but wow, it does all this other cool stuff that may be useful that maybe the pharma companies haven't thought of this as, but because they need new mechanisms that don't do the same stuff that has been going on the last 100+ years.
Right. So I can actually talk about something that, from my lab at UCSF, like one of the final projects that we're still trying to write up as I sort of like shut things down there. Is an observation about antimicrobial peptides in tick saliva. So, and I want to be very clear that like what I'm about to say is not an observation that I originated. I think this has been made by a number of different people in the field, but it really like hit home for me as we started digging a little deeper into this. So the idea is like, okay, you're right. It's not just this like one antimicrobial toxin. There's a whole host of other things. There's like a ton of like predicted antimicrobial peptides. These are like short peptides that are thought to like somehow kill microbes through a range of mechanisms, such as like interfering with their outer surface somehow to penetrating and doing something else. And actually what we've ended up finding is that like some of them are really, really potent antimicrobial like bactericidal activity. And some of them are not as potent, you know, like, I mean, based on the concentrations, we would predict that these are present in the tick saliva. It's like kind of hard to imagine how they're doing it, you know? And so what we started thinking about more is like, has it just been convenient to annotate these as antimicrobial peptides because like bactericidal activity is a very easy thing to assay, right? And that can be a side effect function of them, but like we tend to want to categorize things as having like a single function in biology, and that's actually rarely the case. Most proteins, most molecules can play more than one function. It's us that like desperately needs to label them as something. And so it's actually possible for a large fraction of these, this is my current hypothesis, that like either they're not actually antimicrobial, but under the right conditions in vitro, you can see that, but that they're playing something else, and we've gotten sidetracked with this. Or they are antimicrobial, but there are also many other things that may actually be related to this function. So what we're putting a manuscript on now, which will come out hopefully in the next few months, is that like for a subset of these, we synthesize the peptides that were annotated as defensin-like in tick saliva. But we actually found that they can like cause degranulation of mast cells, like animal mast cells in the skin barrier, human and mouse, and that they can bind and activate these G protein coupled receptors that lead to degranulation in these mast cells. And so we teamed up with this other group that studies this, and they've started seeing the same thing, that like, yeah, like they found that like defensin-like antimicrobials from bacteria actually can cause this to happen. And they think, you know, we think it might be part of a larger circuit of like, yes, maybe it can play an antimicrobial function, but it's also there to like sort of like alert, you know, the immune cells that like there are bacteria around and can do this secondary function. If not, maybe it's even its primary function to do this other thing. To sort of like signal, release more immune molecules, and maybe like even allow for some like longer distance signaling in the skin barrier. Which just forces you to really then, if you're willing to question that, then you almost have to force yourself to question all of it. Like, what are any of these peptides doing? And have we actually found their original function? I think not. Actually, I think that these peptides are extremely versatile. And we don't even know, for example, as we started going down this path, like the way we synthesize it, it can actually form many different sort of like secondary structures. And like, are we even playing with the right one? Or is this something that is itself sort of dynamically regulated in vivo? There's so many questions we still have over these like tiny peptide molecules. And so I think if, you know, and I obviously am thinking about this, like at Arcadia, like, let's say we just like try and like disrupt our thinking on this, like where are peptides found across the tree of life? Like what can we imagine things that they might do? Like, do they play a big role in like behavior, even like regulating behavior of animals? Like, I think there's like a lot more we could think about there and try and approach it in a slightly more unbiased way. And so that maybe that's like one recent insight I've had from my digging around in tick saliva, is that like there's a lot of assumptions we're making about what these molecules to do based on what we can like most easily pattern match them to. But that we may be, sort of like doing ourselves a disadvantage there.
Very interesting. Really interesting. Seemay, transitioning to Arcadia, and you, I mean, it's an amazing portfolio. And I encourage everyone listening and watching to go to the website, and we'll have it in the bio of the show to sort of look at the sort of the scope of the projects. But could you say just a couple words about? I know you've written sort of essays about this, but why did you set up Arcadia? I mean, I know you've written about, okay, you know, there's academia, there's sort of the private biotech space, but there's a lot of other models in between. So say a few words about sort of what Arcadia was all about when you first thought it up.
Yeah. Arcadia basically was about just trying to see from an organizational perspective if we could also evolve something different. That there are a lot of things about, you know, the kinds of science that I think is important to do, as well as the kind of scientist I am, that doesn't perfectly fit under sort of like either model of like industry versus academia. And I think you can take two approaches when that's the case. You can try and like figure out how you're going to like navigate that system that's not optimized for the thing you want to do, and kind of like in between the cracks, like make things happen, right? Or you can actually, if you have like the privilege that I do of like access to a certain amount of resources, then you could maybe think about like, how could this be built differently, specifically to optimize for that thing. And I think like many people often feel in their careers when they're like frustrated, it's like, I just felt like I was trying to like live in a house that was fundamentally not built for me, and that I was spending all of my energy trying to push against that. And that I would almost rather exchange that like frustration with sort of the intense anxiety that comes with building something new. But it's like, it in some ways feels better because it feels like you're like actually trying to build according to the first principles of what you want. And so more specifically, in terms of like the science that I think needs to be done in the world, is that I think there's a huge category of scientists that don't fit perfectly into either, that I've been trying to come up with the right language around this, but it's like a combination between exploration and invention. Like really, it's like people who are very entrepreneurial and want to see like, want to tinker and see that science go towards like applications. And this is something that I think we have a real sort of like difficult time with in academia, because you do need a place for sort of protected basic research that isn't forced to be applied to something necessarily, because that's a very long-term vision of like building up a basic fundamental knowledge that allows for that to happen. And then in industry, you kind of have the opposite of like, well, if there's not a shorter line to application, like we have to be like fierce about just like cutting back on that, right? It's not like endless exploration. But there's a huge gulf in between those. And I think that's like where Arcadia is hoping to sit. For scientists like me who like both, like I do love to explore and I do love to tinker, and I'm fascinated like in a curiosity-driven way about the biology. But the moment I see something that like there's an opportunity for invention, I want to act on it, and I want to act on it unapologetically and with resources and with deliberation, to like see that get done. And that is really exciting to me. And so I think I'm trying to create a space where it's a space specifically for people like that and who like to tinker, but like maybe like, they love evolution, like they're an evolutionary biologist, but they're not quite fit for academia. I'm kind of looking for to create a space for that. So awesome. Then having said that, this is like the perfect area of science, I think, for that, because it's an area of science, like people who want to set up new organismal systems. It's this in this like weird pocket where I think there's immense commercial opportunity there. It's a longer timeline than like most startups are dealing with. So like, yes, it requires a different setup. But if you look back, if you want to rewrite history, like you will see like a lot of the like really like groundbreaking innovations have come from biology. Yep. Like we didn't come up with it ourselves, right? And so I think the potential is there. And then also like weirdly in academia, like this stuff falls through the cracks too, because the amount of work that it takes to build tools and set up a new system, it's like very high risk. It's a lot of like tool development, technology development. And that is really hard when your primary currency for productivity is like journal articles. And more and more, I think we see journal articles are favor kind of like storytelling and these like biological narratives, which is great for some kind of science, but not for what I'm talking about. And so I felt all of these things like firsthand very intensely, because I just started seeing in my lab that like more than half of the work we were doing was never going to see the light of day in journal articles. And I felt very frustrated by that on a couple of levels. Like one, that's information that's not making it out there fast enough. And like the people who could benefit from like all the failed experiments, like, that somehow we're not able to find an efficient way to get that out there. Second, it's like, it's deeply unfair to me that like there's scientists in my group that contribute so much to our ability to move forward, yet they can't capture the value of that because we have such a specific way of like publishing, And so they're forever, I guess, sort of like cursed with being like middle author or what, you know, in that framework, when really they're so core to everything. And it makes me worry about their careers and all this. So I was just like, it's very hard for me as an individual to fix that, you know, like I think the incentives are just not quite aligned there.
Sure.
And I can't like change the system. And there's many reasons why the system is the way it is. It's not all bad.
Yeah.
And so, yeah, those are, I mean, basically the reasons why I started Arcadia is just like I hit a wall. I just felt like, you know, I'm like about to be 40. I'm 40 now. So this is right before I was 40. And it's like, I feel like I have like a, you know, not that I'm going to like die anytime soon, but these are my prime years of like, I have energy. I have like, you know, a desire to really like do something big. Like I either got to like stop complaining and accept the system I'm in, or like do something about it at this moment, you know? So I think one thing that's very important to me, for people to understand, is like, in no way do I see Arcadia competing with academia or competing with like traditional biotech. It is like trying to wedge into this thing that I think is falling through the cracks in both spaces, and ultimately could probably help with a lot of the efforts in both spaces, if the needs are met.
Awesome. Really awesome. Very refreshing to hear, you know, to hear that perspective. But no, it's great. Yeah. So you know, based on that, I mean, as mentioned, the portfolio is broad, and I just thought, you know, we could spend a couple a couple minutes on some of these segments, and I just picked a couple that I find the coolest, but they're all pretty
cool.
They're all pretty cool. And I think the first place I just love to go is sort of into this theme of dissecting the mechanisms of sort of the extremophiles. And you know, this is you know, another just really important topic in the sense, yeah, as you point out, okay, we got these organisms on this planet that love to live in radiation in the middle of the deserts and under the ice caps, and hang out, and are very happy and doing their thing. And you ask questions, right? You know, how do these organisms defend themselves in harsh environments? And then ultimately, what can we learn in terms of the cellular processes, and what can they inform us ultimately on this is a very important theme that we've gotten into a lot on the show, resilience, ultimately human resilience, whether that's me headed off to Mars one day, or whatever the case may be. Talk a little about this. I think in this basket alone, you have, you know, $1 billion company by itself. Take us into a little bit of your interest in extremophile biology.
Yeah. So our interest so basically we had, you know, a scientist joined, Gallo Garcia, who came in with this like deep interest of his own. And I think, you know, honestly, like as a scientist, it's like, there's no greater motivation than the curiosity of the scientists themselves. Right. So like, I try not to be like overly top down within Arcadia. Of course, it's a balance, but it's helpful to listen to the scientists that we interview and like what they're naturally curious about. And so he came in with this idea, actually. And so for the first year, like many other people at Arcadia, we've just been giving people as much freedom as possible to explore, because we don't really know what our like strategy should be, or like what are going to be the most exciting things. And like we've learned a lot in the process of allowing people to just play. Now we're sort of at this inflection point of like, okay, now we've spent a year together playing and like sort of like observing each other. Like we do need to put in some structures and like narrow the focus. And so the hardest thing at Arcadia is this piece, like what constraints do we want to put in place, like after a period of play? Because it's kind of like, I don't think people understand like how stressful it is to have so many options in front of you. It's like, and actually, like I think like most humans, myself included, like walls, you know, like it's helpful to have some walls to push against. And so it's kind of scary to come in and be like, I'm interested in extremophiles and resilience, and I am at a place where I could potentially work on anything in this area, on any organism. And so what we've been trying to figure out is like, okay, let's take a step back. Like, I'm not even sure we don't know. We haven't even convinced ourselves that we're looking at the right organisms yet for what we want to learn. And resilience is a huge category. So let's start mapping out like, what are different modes of resilience that you might expect? There's temperature, there's pH, there's just so many things you could imagine, right? And so that's one sort of like brainstorming strategy. It's just biologically, like a priori, like what are the things that you might even possibly want to learn, and that would kind of dictate like where you look, right? And then the other question is like, what is the upside of learning that thing? Like, given that we ultimately want to move this towards applications, like let's take it like from the totally opposite direction of like, let's talk to people in all sorts of industries, like what extreme conditions like would be helpful to understand more about, and how could that be applied? And we've learned a couple of things like that we didn't think about before, just like cold storage for, sort of supply chain issues. Like this is not something like a basic scientist would sit there and be like imagining, but like trying to like really get into the weeds with like people outside of, sort of our basic research mindset of like, where are the actual problems that are getting solved? And also where are the problems that are not getting solved that like we could uniquely solve? Like we're not trying to get into some rat race that like already a bunch of people are like flooding into some market trying to fix. Where can biology be the shortcut because there's some fundamental thing we like maybe are not understanding, And then also just another insight we've had in talking about this is that like I think a lot of our sort of starting focus has been on how the resilience itself happens. Like what are the mechanisms of like, let's say if we pick on cold as an example, like of like resisting against XYZ during cold temperatures. But actually what's really cool about the system that like Gallo helped write a project page on is the reversibility. That it's not just that they go into dormancy, is that they can go to dormancy in a way where they can come back out. And that's actually incredible, right? Because like a lot of things that we try and engineer are not so reversible. And that like sort of plasticity, I think, is like the asset, really. Like if you, that's what's uniquely different about this biology, is that the organisms are not just going into dormancy and never coming back, that they're coming back somehow. And it's a lot of things that we may traditionally think of like really, really difficult to come back from, like getting into this extremely stable state with like nucleic acids or proteins. Like how are they storing that? How are they preventing any entry into the cell, yet, they're not totally metabolically dead. And so I guess we've like kind of started like evolving in our thinking around this, but this gives you a sense of like how we're trying to approach it. So ultimately you want to like go just as rigorous from both ends of the spectrum and figure out then like, what's the Venn diagram overlap there? And like, we are not yet at a place where there's like not enough things to study, that I think like our scientists need to feel constrained. So the earlier we chime in on the like latter part of applications, the more it actually helps them to like point their laser in a very creative way, so that the process of the research feels very similar to them as before, but like it's very, very intentional of like where are we trying to go with it if it works,
Yeah. No, that's really, that's awesome. And alone the whole, the biologics protection thing alone is, I mean, that's, you win if you do that one. I mean, but yeah, that's really, really.
It's fun. It's like, I mean, I think like as we started doing this more, I mean, it feels very collaborative still internally of like, as I've been taking some of the scientists through this process with me, like, a lot of people coming in feel like worried, like, oh, are you going to like police what I do and force me to do something? And it's like, no, actually it should, if we do it right, it should feel motivating. Like, I'm just trying to give you the tools and information for you to think in a very entrepreneurial way about your own science. And it should be fun. Like, it should be fun when you figure that out, to see like a direct application of that work towards something. Like what would you be interested in if this were your project?
I mean, the thing I've always been interested in this domain, and I spoke to those folks a couple of years ago in the DARPA Golden Hour project with, ultimately if you have somebody that, in the middle of wherever, Afghanistan, and you need to stabilize the life, like our friends the tardigrades and so forth, ultimately how you create that what's the that cryptobiotic capability to go in and come out. Obviously I'm a huge fan of science fiction and long-term hibernation, but that need for short-term hibernation in that golden hour and understanding how that biology works and
Mm-hmm.
how the tardigrades and
Yeah.
everything do it so well. That's always been fascinating to me. I
Yeah.
don't know
Yeah.
how
Yeah.
easy it's
Yeah.
going to be, but it's pretty
I
cool.
don't know either. Yeah. I mean, I think like I guess related to this idea of the reversibility is like I think people
Right.
focus a lot about the proper way to freeze something and not about like the proper way to thaw something. And actually, like, you know, now I've been like learning a lot more about sort of like in vitro fertilization. And so I have a bunch of friends doing like IVF and stuff. And like it's actually the thawing of the eggs that can
Yeah.
be one of the most problematic pieces.
Yep.
So
Yep.
anyways, yeah, I do think like, yeah, whole extreme temperatures are really fascinating.
Absolutely.
And there's so many examples of that around us, you know.
Yeah. Oh yeah. Going from one really cool basket into another, and I think, some of this work actually goes back to your sort of original publications with the sea squirts and looking at at morphodynamics and so forth in tissue patterning. But here you're thinking kind of broader in terms of just basic spatiotemporal movement and properties of cells. We don't normally think about this, but you talk about the dictyostelium, sort of this pond scum that loves to hang out as individual cells, but then likes to get together and in multicellular structures. And it's really interesting because they use the same genes that we do for a lot of aggregation and so forth, and in forming new tissue and morphogenesis and all that. Talk a little about this theme. You call it cell crawling. Again, we think of ourselves. We have these 30 trillion cells or so. We sort of don't think as much about the movement that we sort of here we are. We don't want to think of that cells are crawling around and moving and forming new structures. And when we have wounds and all that, talk a little bit about that, because I think this is another really exciting thing. We don't think, okay, here's some pond scum, and it has a lot to teach us about why we stay as we do the majority of our lives.
Yeah, so like at a broader level, like the cell movement project is really trying to like get at like better ways that we can like categorize microbes through phenotype.
Okay.
That like, right now, one of the main ways we sort of like understand the relationship between different species is phylogenetics, which is helpful for one thing, but like you have convergent evolution, it's maybe a technology that is a little bit expensive if you have to like sequence every single genome of every single thing to understand like how to study it and how to think about it. And so this is like one place where we're starting, where we're saying, what's another way we can look at all the different organisms that we bring in-house and start categorizing them into different like phenotypic categories. And like movement is a thing that is like extremely, there's a lot of diversity for this. And actually like, like what are we even talking about when we say the cell crawls in this way? Like, I think like we have the technologies to get a little more granular with that. There's many different aspects of motility and like starting to group these out in different ways. I think the vision will be to have like not just motility, but like a couple of other categories. This is something that we're starting to coin internally as like quote unquote organismal onboarding. Like every time we bring in a new species, like what are the like 10 things that we want to like collect about them? Like motility is probably one of them. There's probably a bunch of other sort of like molecular, biochemical, and behavioral profiles we want to add to start trying to understand what's similar and different about these different organisms. And so, yeah, we're sort of like trying to push on these different technologies to see how informative they could be. Because what if instead of having to sequence all the genomes of things, you could have like a proxy that's cheaper, more high throughput, that you could just like look at the way they move and be like, oh, I think this is in this category of organisms that might be really useful for us to like study or compare, or there's some insight about this. If you wanted to like think about like actin branching or something at the cell's leading edge, like these are the different things that you might want to be looking at in organisms in those groups. So I think that's some of the big picture goal is just like, yeah, what are more clues that we can start collecting at like a large scale, but ones that are cheaper and that like also we could like crowdsource from people to try and put together like different orthogonal pictures of like the tree of life. In terms of motility, I mean, I personally think like this is a huge opportunity that has like potential applications down the line. Like in your body alone, like there's so many different cell types and they behave differently. They have different morphologies. They move differently. Yep. And like, I think there's increasingly like an interest in sort of developing new model systems for, you know, high throughput screening of drugs that relate to different kinds of like disorders in your body. So like, wouldn't it be cool also like if we learn more about this to better matchmake like organisms with like as like screening platforms for different types of cells in even our own bodies. I think that we don't quite have like as sophisticated of an understanding of how these things match up across the tree with our cells to like do that quite yet, but we're gonna get there, you know. Absolutely. That's where all this is moving, in my opinion.
No, absolutely. Talking about, you know, go back for a minute to the sort of this cocktail theme that we talked about earlier on in terms of, you know, the way nature and sort of evolution works in creating these interesting biomaterials. Yeah, God, it was maybe about 15 or so years ago. I spent a little time with Dr. Ron Sherman at UC Irvine, who you know, is involved in sort of the leeches and the maggots and all sort of what coined biotherapy on basically how you utilize the living organism itself as opposed to sort of dissecting the different components of it. And, you know, they got the leeches and the maggots sort of FDA approved both as this delivery vehicle that was chewing up the wounds, but also the secretions and all that. And there's a lot of interesting strategy behind sort of how you work with a living organism as the therapy as opposed to individual components of it. Any, you know, thought on that? I mean, I know it's kind of a philosophical point at this, but like, you know, if you were to find out that whatever, you know, if I was to swim around in a tank full of sea anemones one day and it had some interesting property that, you know, if we keep studying sea anemones alive doing something, it's better than if you just isolated a certain peptide. Any interesting things that are popping up or findings in terms of what some of these living organisms do for us as opposed to the active biomoieties?
I mean, I definitely think that like we are going to miss out on a lot of cool things if we don't think about it in the way that you're saying, because anytime you have two organisms interfacing, whether it's for a few seconds or like a couple of days, like there's actually a molecular conversation happening. Yep. There's the back and forth, right? It's not like one word gets thrown across and then that's it. So it like oftentimes what we try and do is find that one word and like, okay, an antimicrobial peptide, there, done. But no, there's like an exchange over time of like something happens from one side and there's a response and there's a detection of that response. And there's a lot of like back and forth regulation. And I think a very you know, clear example of this is what's happening in the tick saliva. So over time, the tick saliva, the composition of the molecules, the cocktail of things that are delivered there changes. Sometimes in a very canonical way. Like if you take tick saliva on day 1 versus day 3, like totally different things are happening in the biology. Day 1, it's just trying to build the bite site. Day 3, it's like full blown like feeding, you know. And so there are different molecules at play that it's delivering, right? On day 1, it's very focused on like blocking the immediate response, building that cement-like layer. Day 3, there's, you know, vasodilation agents and stuff to really speed that up, and probably dealing with like a whole different set of immune responses that happens after the first immediate acute response, right? And so I think if you try and study any of these molecules in isolation, you're going to miss it. Not to mention that the things that like are happening at once, the circuits are tied. Inflammation, pain, itch, these things are not happening in a total vacuum from each other. There's like cross communication between those two. So for sure, like I think there's a cocktail effect, and that cocktail effect is like very dynamic over time. There's another thing, which is that it's not just like a one size fits all thing. Like depending on what the tick is feeding on, like depending on the host species, it's also delivering a different set of molecules. So there's this really cool paper from a guy in Texas that looked at like saliva composition of ticks depending on what animal they're feeding on. And it's a different cocktail. And so that's also
paper with lizard feeding that you published. That was kind of cool too. Right? It reminded me of that one.
Yeah. And so I think like you could think of this as like almost frustrating. It's like, like can't quite get a handle, but like actually there's a lesson there. Like
Sure.
there's something that you could like learn from this in terms of like how the tick has learned to adapt to different hosts and like what that means at a molecular level. Like, why would it want to change this? It probably has some molecules that are generalist molecules that like no matter what it's feeding on, it's important to deliver, and it has like evolved to be promiscuous across species maybe, and some that may be especially tailored, which is like why it might prefer a certain set of hosts over others, even though it can sort of adapt if it needs to. So yes, I definitely think that like thinking about things happening as a cocktail, as well as what happens over time, those are both things that like if we can start understanding the complexities of that a little bit more, we may be a lot more effective at like treating things even in our own bodies. And for sure that's going to be true for like, you know, like sensory things or like inflammation, anything where we see like differences in short, medium, long-term regulation in our own bodies. It's going to require sophistication.
Very, very cool. No, I just love I love all these themes. I'm excited for you that
yeah,
you
it's
know,
I'm excited too. I mean, it could end up being a nightmare for us, honestly, as like a, you know, like if we were like a much more traditional startup with like a 2-year runway only on, and by the way, Trove has now merged with Arcadian as like one of our internal projects.
excellent.
It would be really stressful, you know, because that's like basically like your nightmare situation to be like, oh no, like I don't have one molecule, I have 10.
Yeah.
And they have to function together. But I mean, it may actually be that will teach us something fundamental that like is why we don't have very effective therapies for like itch and pain. Like I think there must be something there. If that is like what nature has evolved as the solution, I don't think you can just ignore it,
No,
you know?
no, not at all. No, there's a lot there. I'm just glad to see you, scratching scratching this new surface that people haven't gone. I mean, it's just so spot on, and yeah, I'm excited for for you. So I'm rooting you on here with all these portfolio projects. But what else is coming up in terms of obviously we're getting to 2023 now, but conferences you're going to be presenting at, places that we can hear you give talks, maybe meet you, anything else in the coming months that we should know about while we have you?
Yeah, well, I mean, half of the team is right now at ASCB, Society for Cell Biology meeting. And so they're presenting all their stuff there. I mean, the fastest way to know what we're working on is just to look at our research publishing site. That's like directly linked from our website. Like, it's really funny that something happened for the first time this week that was actually a goal of me and my co-founder Prachi. But like, I didn't think it was going to happen this quickly, which is that I was like, wouldn't it be awesome if there was like such a direct link between what we're doing and what we're publishing in terms of like getting stuff out there quickly, that looking at what the people internally are publishing is like a reasonable way for me to stay up to date on the science that's happening within the org. And it actually happened where someone put up a pub where I was like, I haven't even had time to like schedule a Zoom with this person to like catch up with them on all the details of this thing. So I just read the pub. I was like, okay, now I like know all the ins and outs of like everything they're doing. And I think that's awesome. So like, I think if that's helpful for me, that surely is like the best place for people on the outside to look. We are thinking about conferences next year. I don't quite know what our conference schedule is, but we plan to be very much out there and like to the best degree that we can share what we're working on, because also like our stuff is so early stage that, you know, we would benefit more from being open about what we're working on than trying to like keep it all behind closed doors, because we may or may not want to pursue some IP, you know, 5 years later or something. So yeah.
Got it. Okay. I said, well, we will definitely put the bio and obviously the Arcadia link in the bio to the show, so people can check things out and keep abreast of all of that. One
more thing I was just going to say actually about our like our publications, like Also another great way to interact with me or anyone there, like I read comments, like every comment that someone from the outside puts on our pubs, I read. And there's even been one case where like one of our candidates for a job like commented a lot on a pub, and that like demonstrated how effectively we could like interact and how useful they were, and we ended up hiring them. So
Nice.
also, if you're like interested in like a job at Arcadia, it's a great starting point for engaging with us on the science.
Awesome. Seemay, one thing, last thing while I have you, go to your Twitter site. It mentions biology, Beyonce, and brisket. I love biology. I don't listen much to Beyonce, but I love a good brisket. And I assume, I mean, I don't know. I mean, I don't know if your love for brisket started when you were in Texas, or if this is
Of course.
a Berkeley thing. But
Oh God,
where's the best brisket? I mean, give us some tips there.
Oh man. I mean, basically, like after living in California, I hope I don't piss anyone off with this statement, but almost any brisket you would get in Texas is like awesome to me now. But you know, in Austin, where I'm from, I think, you know, Black's, Franklin's, La Barbecue, these are like great spots. But the key is they're dry smoked.
Dry barbecue.
You know, fall apart. Yeah. And it's in my opinion more pure, because you know, while sauce is of course a part of Texas barbecue, it's like we don't lean on it
Got it.
As much. But yes, I'm a big barbecue fan.
Nice. All right, no, that's important to know. It's an important piece of this whole story. And I was wondering if I get down there, I'll have to check out some of these places.
I could talk. Just ping me if you go. I have much more to say on it, but I'll try and
we'll do a follow-up show on brisket.
Off.
Yeah. Really, really great stuff, Seemay. I just want to, you know, again, I'm going to be rooting you on. For everybody that's going to be listening to this particular episode of our show, across the podcast networks or watching on the YouTube channel, you've been listening to Dr. Seemay Chou, co-founder, CEO, and board member of Arcadia Science, co-founder Trove Biolabs, who's now part of the organization, but a separate website still. Seemay, I want to thank you for taking the time out of your schedule to come talk to us about all these topics. Obviously, thank you for everything you're doing and championing there at Arcadia. And as we like to say on our show, thanks for helping to create a better tomorrow via what you're doing. It's a really, really great story.
Thank you so much. I really appreciate all the thoughtful questions.
Great having you.